All right, everyone, we're going to go ahead and get started. Thank you so much for swimming over here today and dealing with the rain and the wind. My name is Cinnamon Moffat, and I'll be your host for today. Um, and this is a hybrid event, so we have folks online and folks in the room. Folks online, thanks for being here. Just letting you know that your cameras and mics have been disabled for this event. So if you have any questions for us or any questions for today's speaker, go ahead and put those into the chat, and our volunteer Roseanne will read those out and we'll get through those. For folks in the room, you all know this, you've all been here before, but if you have a question for our today's speaker, raise your hand and I'll bring you this mic. Um, before we get jumped in, this is also going to be hybrid for our presenter today. So they are joining us online from Portland. We're just excited that we're able to share this, even with all of the challenges with our weather today. So thanks to everybody for being here. I also just wanted to remind folks that this is our last seminar of 2025. Yay! And so our next seminar won't be until January 15th, when Hannah Glover is a research associate with the College of Engineering at Oregon State University, is going to be talking about monitoring the ocean at the speed of light. New applications for distributed fiber optic sensing. So that should be a really fun talk. Hopefully you can come back after the holidays and join us on January 15th. But for today, we have an amazing speaker, Dr. Catherine D. Rivera, who is a professor of environmental science and management at Portland State University. And for 25 years, since her postdoctoral research, in the Marine Invasions Lab at the Smithsonian, and with Ted, and I didn't find out how to pronounce his last name,
Grosshol,
Cat Grussel.
Grossholtz.
Okay, Grusholz at UC Davis. She has been studying the spread of impact and management of green crabs and other introduced marine species. She is interested in how biological invasions interact with climate change, and more recently, how our language surrounding introduced species affects the perception and management. She also does work on and research on habitat connectivity. And so we're just really excited to have Kat here and joining us. So I'm going to hand the mic off to you, Kat. Thank you very much for being here.
Thank you so much for inviting me to come and provide some recent information about the biology, impacts, and spread of Carcinus maenas along the West Coast. And I'm really sorry that I had to pivot to being virtual today. I thought I was over the flu, but here I am. But I am feeling okay now, and hopefully I won't have coughing fits, etc. Anyway, Hatfield is such a great resource for all of us in Oregon, especially those of us in the marine realm, that I'm really appreciative of being invited here. And thanks for the intro. Anyway, I'm hoping you leave with an appreciation for the adaptability of Carcinus and a sense for its role in the environment and how it might act and interact with your interests, if those lie in estuaries or larval transport or species in the same food web. So I'm going to talk today about why we should care about Carcinus maenas. This is also called the European green crab or shore crab, or most recently, especially here in Oregon, five-spine shore crab. Um, and we'll look at their population growth and spread, and then some impacts we expect them to have. A little hint towards population growth and spread. You can see in these pictures, females produce thousands, hundreds of thousands of eggs, up to 2 million in their lifespan across 5 breeding cycles, and actually 5 years, up to twice a year. And their populations can grow. We often get lots of green crabs or Carcinus in our traps. And we'll look at impacts later. First, how to identify them. They're often called green crabs, so people expect them to be green, but they're often this olive color. Sometimes they're orangish or reddish or other colors, and so it's not, and of course we have other greener crabs, so it's not a great descriptive name for them. Some people are calling them five-spine shore crabs because they have 1, 2, 3, 4, 5 spines on either side of their eyes in this fan shape. And that combination is pretty unique on this coast. So in this map, we can see where they hail from. They originated from northern Africa up to northern Europe. And so their temperature tolerances are enormous and they can survive in all these waters. And that has helped them survive in all sorts of places we have introduced them to, unintentionally introduced, including Australia and Tasmania, South Africa, South America, and the east and west coasts of North America. I'll also point out here that when this map was made long ago in 2021, their northern range limit was in about Vancouver Island, and it since has expanded, which we'll look at a little more detail here. And when we look at the West Coast spread, they were came to the west coast of the United States from the east coast, where they've been for about 200 years. They were first found in San Francisco Bay, really low numbers in 1989. So it seems like it was a recent introduction then. And quickly spread up the coast, arriving in Oregon by 1997. And then they kind of stalled out and then had a couple more jumps since then up through British Columbia and finally a couple years ago into Alaska. Dr. Sylvia Mata from OSU has found that this spread is really correlated highly with El Nino years and with warm winter sea surface temperatures, a late start to upwelling, change in timing of the spring's transition, abundant subtropical copepods, and high Pacific decadal oscillation values. And all these, when these co-occur, we get green crabs spreading north. Towards the northern edge of the range here, there's a team in the Metlakatla Indian community that's been surveying for and removing Carcinus from the waters of Annette Islands, which is where they got in 2022. And since then, they've spread just a bit further. And you can see that the catch per unit effort here is really much higher in 2025 than it was in 2024, which is this blue, really small line here. And they removed this summer over 400,000 Carcinus from the shores of that island, way more than before. And that is really suggesting a quickly growing population with a lot of potential to spread further. Is there high risk of further spread? I would say yes. These crabs, the adults walk around. They can swim some. They're portunids, but they don't quite have the paddle foot of blue crabs and other portunids. But their larvae are planktonic and are transported by currents, hence their spread on El Nino years. And the larvae have broad environmental tolerances, the adults even more so, withstanding temperatures from 0 to 35 degrees Celsius and salinities from 4 to greater than 34 parts per thousand. So they live just fine in marine realms, and also they're a true estuarine crab. We did some modeling a little while back, looking at where they could go worldwide, but here focusing on Alaska. You can see that pretty much all of the Gulf of Alaska has habitat that's suitable for Carcinus to live should they get there. And this was done with environmental niche modeling, looking at salinity, temperature, chlorophyll, and a bunch of abiotic variables. And then if there's 2 degrees warming above the 2011 numbers, and there's been some of that warming already, then they could get up to almost 63 degrees latitude, kind of clearing Alaska's beard there. Does this matter to us in Oregon that they're spreading? I would say yes, for a couple reasons. One, we now have some local recruitment. Until now, all our recruitment has been from the south. And then now there's this possibility of recruitment from the north too. And with recent northward expansion, the genetics has shown that there's now in Oregon and Washington some recruitment from the north. So across local recruitment, recruitment from the south, recruitment from the north, this all suggests that we might have more stable input of Carcinus into our system. and allowing populations to grow and thrive. We did some earlier modeling also where we grew larvae or cultured larvae at different temperatures and temperature combinations, and then projected that along with sea surface temperatures on the East Coast and West Coast. Not surprisingly, on the East Coast, everywhere they already lived, it showed that it was good, suitable habitat for them. And this was with 2006 data. And of course, it's warmed since then. But in 2006, it showed a lot of the West Coast sites at the northern and southern range, parts of the range were suitable, but that actually Oregon waters were kind of cold for Carcinus then. And it was not predicting it as a great place. And we're going to look at this in just a second. But first, I'm going to also note that we showed back then with this other method that also much of the Gulf of Alaska has had suitable habitat already in 2006 then. And then looking with some warming, this is aligned with niche modeling that most of the Gulf of Alaska is suitable. This shows a little more cool, unsuitable habitat due to temperature than the niche modeling did, but it's also earlier data. Okay, so let's look at Oregon for a second here, where in 2006 we thought it was too cold for them. And here's data put together by Sylvia Yamada and Sean Skula and others who have been trapping Carcinus along the Oregon coast for decades. Sylvia started this as soon as they arrived. And you can see their abundance here on the y-axis across years from 2002, and noticed that there just weren't many green crabs at all in Oregon's waters until about 2016. And even that year was pretty low, with around like half a crab per trap being caught. And then starting in 2016, the numbers really start to rise. And now, and this is Coos Bay, one of the more invaded bays by this crab. Now there's over 7 crabs per trap, which doesn't sound that much, but we're trapping in all sorts of places that aren't great for green crabs also. All right, so this isn't really out of line with what our model suggested. It has warmed some in the interim, and that might be one of the reasons why their numbers are increasing now. And then also with the migration north, there's just more possible seeding. This is a bit of a side note, but I think is super interesting. We did some work with Carolyn Teepolt, who's a geneticist, and this was work that was done in a California lagoon that we were studying, which is on the right side, and all these other sites from Elkhorn Slough in Central California by Monterey up to British Columbia. And you can see that, and this graph is a non-metric multi-dimensional scaling graph. So points that are close together are more closely related to each other in the genetics of the crab. And you can see that all these points are kind of clustering together. This blue here, these are all these points from this one enclosed man-made lagoon in California. And it's just different from others. We were studying that because we were doing an eradication experiment, which totally failed. They overcompensated and we ended up with more green crabs, but that's an aside. And interesting to the point of this talk is what's happening on the left here, where you can see that the sites from Elkhorn Slough, the southernmost sites here, and up here we have the northernmost sites and sort of Oregon in between in the blue here. And what you see here is pretty much a latitudinal separation of along this axis, and a temperature separation, really. And so what these data suggest is that Carcinus may well have the capacity for rapid adaptation, including response to temperature. They have standing variation in the genome that allows it to respond to temperature. And that work was done earlier by T. Polton, Samara, and Palumbi. and that they also have SNPs, single nucleotide polymorphisms, associated with temperature and cold tolerance that are clustered together on their chromosome in a place that doesn't have much recombination. And so there's like this inversion polymorphism in with genes in linkage disequilibrium that has allowed for this rapid temperature adaptation, which is, I think, just super cool. And so even though larvae are going from place to place quite quickly up and down the coast, there's still this temperature adaptation. This might be one of the reasons why this is such a successful species all around the world where it goes. Not only is its native range spanning all these different latitudes, but then it can also quickly adapt. All right, so that's what I had to say about population spread and growth. These things are happening quite clearly, and therefore a potential issue for us here in the Pacific Northwest. I also wanted to look at and share with you some research we've done on impacts that we expect them to have. You can see this Carcinus here eating Hemigrapsis oregonensis, our native shore crab here called Oregon, our hairy shore crab. And there's been some research done over time that shows that they greatly impact their populations. Here we can see the number of those Hemigrapsis oregonensis or hairy shore crabs on the y-axis and the number of Carcinus on the x-axis. And you can see that where there are few Carcinus, there are many shore crabs, and then that those numbers decrease as there's increased in Carcinus. We went on to show in that paper that it's directly because of Carcinus eating them, and which they did in lab and tethered in the field, and that impact persists across years with a fewer Hemigrapsis over time, but especially when they're on years where there's more Carcinus. All right, anyway, we know from that and other work that this crab preys on a diversity of taxa, and including fishery species. Work on the east coast of the United States, as well as out here, have shown them eating clams. They're blamed for the collapse of the soft-shell clam industry in Maine, for example. Just a whole variety of different clams, too. Oysters, which I'll talk about later, crabs, which I'll talk about later, and that they also outcompete native animals, including Dungeness crabs. Some work by Sean McDonald up in Washington has showed that. And that they can impact eelgrass as well, which is, of course, a real key habitat for fishery species. Shown here, you can see a little early benthic phase Dungeness. You might be able to see its legs coming around here on this eelgrass, and it just rides the eelgrass up as the tide comes in the estuaries, taking these little crabs away from benthic predators, potentially. All right, we'll come back to that later. So I wanted to talk about some work that my lab has done on oysters and Dungeness crabs in eelgrass. Starting with oysters, this is work done by my finishing master's student, Isabel Mies, and we know that Carcinus eat and even prefer bivalves, especially oysters. And Carcinus aren't that big of a crab. Like a big one is pretty much this big. So they're really smaller than Dungeness and red rock crabs and Pacific rock crabs, et cetera, which could theoretically eat lots of bivalves and do. But we wanted to know how green crab impact them relative to the native crabs. We also know that oysters really are important at ecosystem function, right? They filter a lot of water, clearing it of algae and small particles as they feed themselves. So we were interested in how Carcinus impact not only oysters in their survival, but also the filtration rates of oysters and how that changes with environmental gradients. We did this work partly in Netarts Bay and partly in the lab, and it really stemmed from our sampling in Netarts Bay. And you can see here, the mouth of the bay is up here, a little covered up, but we sampled in all these sites in green from towards the mouth of the bay till, towards the head of it or sort of mid-bay. And let's look over at the graph on the left here where we have the percent of crabs that could eat oysters, not the little ones, but like the cancer crabs and green crabs, et cetera. The percentage of our catch and traps of these crab eating or oyster eating crabs that were green crabs. And you can see that in these sites that are by the mouth over here, about a quarter to half of the crabs we caught were Carcinus, and the others were different cancer crabs. And then as we get farther into the estuary in the less marine areas, even in this very marine dominated estuary, that proportion increases until all the crabs that we catch are green crabs. And then the number of crabs caught are on the bottom too. And you can see that the crab numbers are higher farther up estuary as well, because the green crabs do just fine there. So that's the pattern through the Bay of Crabs. And then if we also look at where there's oyster aquaculture, so there's this great shellfish hatchery here, and then a fish hatchery up here, and those are a bunch of oyster leases are in this area. And also there's just this really productive shellfish area where people can go out and get clams and cockles to eat. And it's in these areas that we find a lot of green crabs. So that led us to this hypothesis that Carcinus or green crabs are going to have a bigger impact on oyster survival and ecosystem function than the native crabs. And with the ecosystem function measured in clearance rate. And we found that expected this because one, they're in a different part of the bay, they're overlapping with oysters more. And two, we know they prefer these. And we also expected they might have a greater impact in warmer water because of the really broad environmental tolerances of Carcinus. And also, with warmer water, metabolism should increase, and therefore they should be eating more. So, Izzy conducted this experiment in lab where she looked at the 14 degrees Celsius water in tanks. And she picked that because that was the temperature of Netarts Bay at the time of the start of the experiment. And then 4 degrees warmer than that, 18 degrees Celsius. And it actually has been getting to 18 in the summer, and especially in Back Bay areas. So this isn't crazy warm or anything. And for both these temperatures, looked at four different predation settings. One that just had oysters, no crabs at all, and then a setting of two Carcinus, and a setting of two Pacific rock crabs, Romelian antenarium, formerly Cancer antenarius, and then also in tanks that have one of each crab. And then had 7 trials per tank of all these different 8 combinations, randomly dispersed or assigned in the lab. And we looked at this across 3 different phases. First, the acclimation phase, where the oysters get put into the tanks, and there's the crabs are in there too, but they're in containers. And so their effluent is in the water, but they're not at all physically interacting with the oysters. And so that's the acclimation / olfactory cue phase. Then in the next phase, which we're calling a non-consumptive phase, she put mittens of sorts on the crabs so that they could walk around on the oysters but not eat them. And I should say that these are oyster spat, so they range from like tiny little oysters up to about 30, 40 millimeters. So they're small. Also mentioned that there's an OSU student there at Hatfield, Brendan, who is looking at how like what sizes can be eaten by green crabs and the interaction between green crab size and how big oysters they can eat. And boy, green crabs can eat a lot of different sizes of oysters. That's really cool work. Um, but an aside at the moment. So they have this non-consumptive phase, and then the third phase is a consumptive phase, in which the mittens are off and the crabs can eat the oyster spat. We measured clearance rates, I should say that Izzy measured clearance rates. I did none of that work. And just measuring the algae, she put in shellfish diet to begin with and looked at the concentration at the beginning of each of the starts of the phases, each of which were 2 days, and then at the end of the experiment. And also, of course, looked at how many were preyed upon. All right, so what did she find? Here you can see that the mean number of oysters that were consumed per crab treatment. And you can see that green crabs ate the most at over 3 oyster per tank, and that the least were eaten when you had one Carcinus and with one rock crab. We expected this because rock crabs can prey on green crabs and do. And Sylvia Yamada and Hunt had done some work a couple decades ago in Yaquena Bay looking at the distribution of red rock crabs, Cancer productus and Carcinus, and also looking if there was and they found reciprocal abundance. And then they also looked at the mechanism behind that and did find that red rock crabs prey on green crabs, which we've seen since as well. So we were hoping to use red rock crabs, but they actually were not very abundant in Neidhart's Bay this year. So we used their close relatives, Pacific rock crabs, instead. It may have been just too warm for the red rock crabs. They're really marine, cool water crab. Anyway, so green crabs eat the most. Then we were looking at clearance rates to check out the ecosystem function impacts of Carcinus. And here you can see on the left side, we have the cooler water, 14 degrees Celsius, and we'll start walking through that across each of the different phases and different crab treatments. And you can see that, well, just looking at the oyster only, they're clearing a good amount in these tanks across temperatures, across phases. And then if you add crabs, they are decreasing how much they clear. And they decrease most in the acclimation phase if green crabs are there. And it's a huge decrease here at 14 degrees Celsius. They're going from about 22 liters per individual per hour of water being cleared to down to about 7.5. So it's a third of the amount just from having green crabs in there, not even interacting with them. And that there was also decreases from when there's other crabs too, the rock crabs, but not as much as if it's two green crabs. Then once the crabs can walk around, there's even greater suppression of clearance rates. And then by this in consumptive phase, we decided to use the clearance rates per surviving oyster at the end, and so there's probably some extra clearance by oysters that aren't counted here, and so those rates go up a little bit. But regardless, they're low across all 3 crab treatments. Comparing that to 18 degrees, mostly it's the same trend of green. If there's green crabs in the tank, Carcinus in the tank, there's greater impacts suppressing clearance rates, except for weirdly here in the acclimation phase when there's like no impact at all measured. That could be because elevated temperatures promote the chemical Q degradation of Carcinus. Not sure about that at all. But other than that, there's this pretty clear trend across temperatures, across phases, that if a Carcinus is in the tank, there's impact to the oysters. All right, so in conclusion, Carcinus totally eats oyster spat, as you can see in this picture where there's bunch of broken shell from them consuming oysters. We found that Carcinus ate more oysters than rock crabs did, and that consumption is lowest in mixed tanks. That crabs impact both the Antinarium here, the rock crab, and Carcinus are impacting oyster clearance rates, even when they're not consuming them at all. This is a pretty big deal because if you have a bunch of Carcinus there and they're crawling around oysters, oysters aren't feeding themselves and aren't helping clear the water either. And so, if they're not filtering the water, that could have an ecosystem impact. And of course, they're not growing themselves. So, even without eating them, they're having a pretty big impact from their effluent, from crawling all over them, etc. And then we would expect a much bigger impact, not only because they're eating more, but because they're actually living in the part of the bay with the oysters. All right, moving on to another favorite in Oregon, Dungeness crabs. We already know that Carcinus will consume Dungeness. There's some good work from Sean McDonald that showed that small Carcinus ate about 10 to 15% of the early benthic phase Dungeness in structured habitats in the field. But at that time, crabs didn't interact that much. Just there weren't that many green crabs in the Pacific Northwest. And so we wanted to revisit this and also examine the roles of temperature and salinity, as both are expected to change with changing climate and, of course, change on estuarine gradients as well. And we see, as we just discussed, that there's green crabs throughout the estuary. So we wanted to see the effect of temperature and salinity on Carcinus predation on Dungeness, and hypothesized that due to the greater tolerances of Carcinus than the juvenile Dungeness, and due to the increased metabolic demands for higher temperature and lower salinity, that we'd find increased predation on Dungeness in these high temperatures and low salinities compared to lower temperatures and higher salinities. So, and this work was done by Trevor Gelling, who's an undergrad in my lab. And so he looked at this in aquarium tanks across three temperatures, 16.5 up to 20.5 degrees Celsius, across two salinities, pretty much marine, 32 parts practical salinity units, and 22, so like true estuarine salinity in these experimental tanks. And the tanks were covered up, so other than when we made observations, they wouldn't affect each other. You can see here there's some structure in the tanks, and that the juvenile or early benthic phase Dungeness used that structure to escape predation. These tanks had a lot of flow in them too, and the Dungeness spent an inordinate amount of time just like swimming up in the flow, avoiding the Carcinus, or it looked like they were avoiding them, but nonetheless, some were eaten. We also fed the crabs before the experiment and during it, giving them chunks of tilapia throughout, and we put in 4 mussels. And so there was a lot of food for the Carcinus in the experiments. It's not that interesting to know what starved crabs eat. So what we found is that they ate the most Dungeness in marine water and cooler water, and or sorry, like marine salinities and cooler water. And so you can see over here the more marine water, and there's just higher up top is they ate the most, and down low they ate none. And so they ate more than over in this other treatments, and also that's true across salinities, the left side compared to the right side. Sorry, the temperature was the left in the blue here. And so there wasn't a temperature effect by itself, but there was a salinity effect and the interaction between temperature and salinity. So we know that they are eating them. It's exactly opposite from what we predicted. We thought they'd eat more in warmer temperatures because of metabolic effects and differential stress, and instead they ate less. And then also we thought in the lower salinities they'd eat more, and you can see they did not. So, I mean, they do live on the outer coast, on the east coast of the United States and Canada, and also on the other side of the Atlantic. They only live in estuaries on this coast, possibly due to high wave action and energy of our wonderful Pacific Ocean, and possibly in part due to predation by more fully marine creatures. Regardless, in these tanks, and despite the different temperature salinity combinations, the crabs ate all four of the mussels given to them in the 48 hours of the experiment, and they ate two-thirds of the Dungeness in the tanks with them, often eating the Dungeness first. So here's another picture of one eating it. There's this question about, okay, well, does that translate to the field at all? Here we know that Dungeness are in structured habitats, possibly escaping benthic predation, etc. So we wanted to look at some field data too. And these data are from Sean Schooler down at South Slough and Sylvia Yamada and a bunch of their colleagues. Over time, Sylvia, as I mentioned before, spearheaded a bunch of trapping throughout Oregon for Carcinus and trapped across 10 sites in Coos Bay here from 2005 till 2025. And what you can see here is that there weren't many green crabs early. We already knew that until about 2016. And then green crab numbers really increased at some of these sites. And in sites where that happened, there are really low numbers of Dungeness crabs and actually red rock crabs too, which are shown in purple here. And so this is correlational field data that's in line with the tank experiments, that it could be due to predation by green crabs on Dungeness crabs, that there's just few Dungeness in the field. And this has also been found, and even more strongly up in Washington, they found huge decreases in Dungeness where there are many green crabs. I also want to point out these four sites with stars because they're in the next slide. So at these sites, almost all the crabs, if not all the crabs, at those sites are Carcinus, green crabs. And so we wanted to know if we tethered up Dungeness, and were about 40 to 50 millimeter carapace with Dungeness, if they'd be eaten in the field. And so Sean and company went ahead and seined and got a bunch of Dungeness and tethered them and deployed these tethers. They tethered them benthically, so, and you can see they have kind of short leads, so they can walk around, but it won't get too tangled up. And they can bury as well. So they're on the bottom where most green crabs are. They also tethered them about 15, 20 centimeters up off the bottom to make sure that the predation on them is mostly benthic, to suggest that those are crabs, since these are like the only crabs at these sites are green crabs. But what if it's something else eating them? In fishes, then you'd expect the upper ones to also be eaten. And also with the idea that they might ride up on eelgrass to escape predation. All right, so what do they find? You can see that that the suspended 30 centimeters up, I think I said 20, 30 centimeters up crabs, tethered Dungeness, were barely eaten. At most, they were eaten about 40% of them. And the benthic crabs that were eaten much more. And I should say, tether is just like a little leash that holds the crab there, so we know that it didn't just walk away and know that it was eaten instead. It's like I always picture the goat in Jurassic Park that gets eaten. It's on a tether. So, eaten by in that case, T-Rex, in this case, Carcinus maenas. Anyway, so the benthic ones, you can see that the percent of tethered eaten is low. Most of them are surviving here. on when Carcinus numbers are low, but that proportion surviving decreases until zero, where they're all eaten when we had 30 Carcinus per trap being caught. So really high densities and actually pretty heavy predation also at 20 parts per thousand. So even though these crabs are, they're not at all full size, but they're a decent sized juvenile Dungeness, rivaling the size of a lot of Carcinus, they're still getting in these sites that are the only crab around is Carcinus. All right. So in conclusion, Carcinus ate two-thirds of the early benthic phase. These were like 10 to 20 millimeter carapace width. Dungeness and lab aquaria, despite having other food. They ate more of the Dungeness in fully marine and cool summer water conditions, against our hypothesis, that they have reciprocal abundances in the field, and that tethered Dungeness were eaten more at sites with more Carcinus at them. I will note that there's been some work that shows that in Oregon, not so much in Washington and Alaska, but in Oregon, a large proportion of Dungeness recruitment is not in estuaries. And so maybe there's less of a threat to this fishery here in Oregon, or maybe not even much of a threat. That's certainly what ODFW is expecting. Washington and Alaska are more worried about the potential impacts on Dungeness recruitment from green crab predation. All right, transitioning into impacts to habitat. We know that eelgrass offers important habitat for many species. Eelgrass restoration projects have failed in areas with many Carcinus. Of course, a lot of eelgrass restoration projects have failed, period. We have a lot to learn in that space still. And we also know that eelgrass has decreased in some places with a bunch of Carcinus. But in other places, we have a lot of Carcinus for decades and plenty of eelgrass. So there's experimental work that has shown some impact in enclosures. Brett Howard up in Vancouver showed that Carcinus density affects eelgrass in enclosures with one crab per meter square not having much of an effect, but four having large effect. But we're still kind of wondering about like, why is there this variation in the field across sites in whether eelgrass, Ostra marina, does fine with Carcinus or not? So I'm going to talk about two experiments here. One that was done by a former master student of mine, Kim Brown, where she wanted to know if eelgrass density affects how much it's impacted by Carcinus. And so she looked at open plots as procedural control, and then enclosures shown in this photo here in Neatharts Bay. Enclosures that did not have crabs in them or that had two crabs in them. These were half meter square. So this is at that 4 crabs per meter square density. And looked at them with, yeah, 2 or 0 crabs in them and 8 replicates per treatment combination. And I forgot to say, we hypothesized that there'd be more impact on low density eelgrass because there's just less rhizomes there. And so it's easier for theoretically easier for crabs to dig through those areas to get clams, etc., if eelgrass density is lower. So what you can find here, you can see in the graph to the right that shows the change in percent cover of eelgrass in these low densities, so like 40 to 60% cover, medium 60 to 80, and high density 80 up. Enclosures that were closed in the gray or in dark green or open in the light green. And you can see that in the open enclosures, there's like no decrease, no net decrease, and sometimes even a very slight increase. So there's probably a little bit of enclosure effect. But when no crabs were in enclosures, or that is when Kim did not put any crabs in the enclosures, there wasn't much decrease in eelgrass at all. There's some, and occasionally. It did turn out that there were two red rock crabs in one of these enclosures, and that could explain some of this. But then when there's green crabs in the enclosures, that there's definitely a decrease in eelgrass, and that this is especially true at the low density, although that effect was not quite significant, 0.08 for that probability. Here's just a photo of a medium density eelgrass. Actually, that might be low before and after the crabs had been in there. This is a 2-week experiment. So they're really ripping up a lot of eelgrass in these enclosures. All right. Then current PhD student in my lab, Kyle Swanson, who's there in the audience, um, wanted to follow up on all this eelgrass work, looking at if temperature is going to interact with Carcinus presence to impact eelgrass. We hypothesized that given the increased metabolic rate that comes with temperature, that Carcinus will have greater impacts in high temperature tanks than in lower temperature tanks. And he set this up so that there's ambient water. This was done at Hatfield. Thank you, Hatfield. Ambient water coming in from your Quinn Bay, or temperature warmed beyond that, 4 or 6 degrees Celsius higher. And that's crossed with having 0 or 1 adult in these meter square tanks. So you'll remember that I mentioned before, having one crab in a tank didn't have a lot of impacts compared to 4 in a tank. But he wanted to make sure that this is kind of realistic out in Yaquina Bay. 1 crab per meter square is like decent expected density. We do not expect 4. And in addition, he wanted to be able to find out, well, if there's impact, is it higher with higher temperatures, which again we expected, and didn't want just all the eelgrass destroyed like you would have with 4 crabs. And so he has this temperature by crab presence or absence interaction in 10 tanks per treatment combo. And these were in flow through tanks. And you can see that there's plenty of clams there in the sediment that he hauled into the tanks. And then also brought in eelgrass and grew it. And then there's the inflow and outflow and hobo measuring temperature. And then, of course, the green crab or not. And this is just a photo of what the tank actually looked like. Here's an experimental timeline that shows that there's a week acclimation time where eelgrass is growing and some clams are additional. There's already some clams in the sediment, and then Kyle supplemented that with more clams and warmed up the tanks, added green crabs. At first not interacting, but then finally during this experimental week interacting with everybody. And then just in case a bunch of the clams were being eaten over time, added some more clams at day 6, and then ended the experiment on day 8 after the start. So what did he find? You can see that first, where there are no crabs, not many blades were missing. So green crabs had an impact on eelgrass loss, with only loss really where there's green crabs. That's not surprising. And then, of course, there's this real question about temperature. Does temperature affect how much they're eating and or destroying eelgrass? Now, green crabs do eat eelgrass. Mostly juveniles do, but they destroy it in digging up clams. And so that's thought to be, and these were not juveniles, these were adults. And so that's thought to be the major impact here, is just digging up the blades. And you can see that there's some increase in warmer waters, at least over here, but not so much over there. And that other than crabs, the real main effect that was found, it's not temperature, but it's adding more clams. When Kyle added more clams at that 6 day mark, you can see that this is the mean blade removal rate per day. This is in the initial time, and it's really low, and then that really increases once more clams are added. It's still not particularly high, though. It's still like half to maybe 3/4 of blade removed per day for the one crab. Of course, this would be higher if there's more crabs in there. Um, There has been some notes in literature suggesting that having just available food uh, encourages the Carcinus to dig more in eelgrass and create more disruption. And now here's experimental evidence that goes along with that supposition from some earlier papers. We haven't yet gone back and figured out from all the other literature in eelgrass about clam availability. Not sure that that will be available there. Um, but clams are available throughout, but they were really dug in early in this experiment and less so later. All right, so in summary for the eelgrass findings, and where am I on time? Oh, I'm talking too much. We found that Carcinus, we wanted to know if Carcinus presence influenced the amount of seagrass uprooted, and the answer is yes, though only modest amounts removed in tanks when there's only 1 crab per meter square, more in the field enclosures. We wanted to know if eelgrass density affects crab impacts. Maybe, not strongly, but the most impact, though not significant, was at the low density of eelgrass. And if warming accelerated eelgrass loss, and the answer is temperature did not in these experiments impact Carcinus foraging rates at low crab density here. And it didn't, there was not interactive effect between Carcinus and temperature either. I'm going to really quickly do this one and not show the videos. So my other student, Jen Riddle, also a master student in my lab, just did a pilot study on predation on herring roe by Carcinus. And you can see that if there's just roe, all of it was eaten by Carcinus. And if there's other prey available too, there are some hairy shore crabs and some big ghost shrimp and some bent-nosed clams in with the herring roe in this case. And in that case, about 2/3 of the time roe was eaten. It was always eaten solo, often otherwise. In about 1/3 of the trials, crabs ate only roe when other prey was available. So they sometimes really preferred it, and all size groups ate it. I was going to show you some videos, but I'm way behind, so I'm not going to, just the munching on roe. And so Jen did a pilot last year. She's going to be working down in Yaquina Bay and Hatfield this March. So look for updates on this. After that, she's going to examine in their preferences and look at the nutrients of the different food, climate influence. She's doing a lot. And the impacts of Carcinus relative to the impacts of Dungeness, because they both have been seen eating roe in the estuary, and also the spatial overlap between roe and different crabs, like we were looking at in Yaquina Bay. I mean, in Netarts Bay. All right, here's another video. Got a lot of fun videos of crabs eating roe. In some. So there's been some work looking at the economic cost of Carcinus back in 2001, a couple decades ago here. Pimentel did this estimate that about $44 million a year is lost due to crab predation on all these different bivalves. A little more recently, Ted Grossholtz here, shown on the right, who's in Carcinus, did an economic study and found that really losses have been minor, but did note that they increase significantly if densities increase, has been happening in Oregon, or if they spread into Alaska, which clearly has happened as well. All right, so we expect them to have all sorts of impacts to species near and dear to our economies and culture. And I don't want to leave us on a dark note here. I think there is some hope. Lots of species eat Carcinus as well. Gulls have been shown to. We did some work in on the East Coast showing that blue crabs, Callinicus sapidus, eat them, setting their southern range limit. The crabs on that coast. As I mentioned before, Hunt and Yamada did this work on Cancer productus showing a lot of predation, though not as much as Callinicus does. Otters do too. There's been some observations of river otters eating them, and work we did in California showed that sea otters have kept numbers of Carcinus down in Elkhorn Slough, which is a sea otter hotspot. And of course, people can eat them too, and do. They are a lot of work because they're not as big as, say, a Dungeness, but it's a food source, and there's that if you can't beat them, eat them strategy out there. And I'm going to throw out there just a maybe. Might it be useful to have this resilient crab as climate change impacts the oceans? Maybe, or maybe they're just going to create more impact. Want to also mention mitigations for a moment. Oysters can be grown up from the bottom, and as you saw, like with the Dungeness tethering results, and I didn't show you this, but Izzy also tethered oysters low and high. They're not really being eaten high up that much. So there's a bunch of oyster culture that already does that. That can really decrease crab predation, which could be important. We met oyster growers who called just their heaps of shells crab hotels for green crabs. Um, and then, of course, there's probably a great idea to trap in key areas to protect resources. Washington's doing a big removal experiment throughout the state, but we probably need to focus on protecting resources and then maybe promoting predators. That's a super controversial statement that I'll leave you with. And also just want to thank a ton of folks who helped with all the different pieces of data here. And I do want to mention the funding from the Mammy Markham Endowment Award to Kyle to work on eelgrass and Carcinus. And thank you all for listening. I have a couple minutes for questions.
Wonderful. Thank you so very much. So one fun thing happened that you might not have noticed is that here at Hatfield we lost power and we're running on a backup generator, but we've got you back. So there was a little gap right in the middle of Kyle's presentation part of that slideshow. But I think we'll just transition into questions. It might also mean that Roseanne lost questions. If you had put in questions during the talk in the chat box, you might need to put those back in. But for folks, do you have any right now, Roseanne, questions online? Okay, go ahead.
And is it known how they were introduced to San Francisco Bay?
Um, yeah, it's thought that that's probably from bringing over aquaculture. They've been found in often aquaculture is transported with rockweed, and that's a habitat for Carcinus on the East Coast, and so it could have just come in with there was an aquarium there where it was found just outside. So whether it was aquaculture or for that aquarium. It's just transported from the East Coast that way. And not many. Like there's estimates that it was something like 8 to 15 crabs, and that this whole population on the West Coast is from that really small introduction.
Interesting. Questions in the room. All right, I have Steve here.
Hi, Kat. Steve Rumrill here. Thanks
Hey,
so
Steve.
much for this wonderful summary and for all your great work moving from the science into facing the difficulties of management of the species in Oregon. I have a particular question. Early in your presentation, you showed the map of the distribution of green crab in Netarts Bay. And if I recall, there was a huge difference between your site number 5 and number 6. Site 5 and lower numbers were more marine, I think, and 6 and such. But
Yep.
that looked like those sites were maybe less than, 200 meters apart. What's going on there between site 5 and 6?
There's a freshwater input right there. It's the by kiosk one. I don't know. I'm guessing you are familiar with that, but there's an outfall pipe with freshwater input, and there's just like a lot of green crabs there. There's like it's a place where I go and I'm like, I need 100 green crabs, and I'll go hand collect them.
Okay, thanks for that. And then the other question was about Kyle's work, so we'll press on that too. The real issue there is using the information that you gathered from the oyster interactions. Of course, the native clams are buried, soft shell clams down quite a ways, manila clams up near the top, the purple varnish clams up near the top. So I was wondering about just any of the inferences that you may be able to make about how deep the crab are digging in search of prey.
That is a great question, and I'm going to let Kyle answer that.
Um, my answer would be not very deep because those initial clams I put in, and I was only doing like up to I think 20 millimeter valve length because they just couldn't handle much bigger than that. And even in that like 5 day acclimation period before the crabs were added, it didn't seem like well, and from the data that I got, it didn't seem like they were able to see those clams or sense them or really dig for them. They were too deep. And so it was really when those second round of clams got added, when they're right at the surface, like freshly added, and they're really close to the surface, that the crabs are able to either sense chemical cues or access them or some combination of those two. And so that I think that's like maybe 10 centimeters or less of sediment is really where they're able to forage for clams.
And
All
that's
right. Any other?
definitely consistent with what I've seen too. Thanks, Kyle.
All right. We've got one more question online. Go ahead, Roseanne.
Is DNA early monitoring shown to be successful for monitoring green crabs? I see this is used a lot for zebra mussels, but was not sure if this was used on these.
What a great question. Yes, we totally rely on eDNA for mussels, but there's been some work, I think by John Darling, that suggests that crabs just don't slough off DNA in the same way to make eDNA monitoring work as effectively. So Washington is starting to look into this. For detecting Carcinus in their shores, but and maybe Alaska too. But the thought is that it's probably not going to be effective. There's some work earlier that was trying to really start up early detection protocols with that, but it's probably not going to be a go-to.
Great. I had a question from me actually, and we can wrap up here real quick. You had alluded to language in your bio, so I was curious about invasive, non-native, and all the different languages that each of the different states uses, and the impact of that and management, and your thoughts there.
Yeah, we just did a survey, and if any of you took it, thank you. We're about to submit it, I think, on Monday, um, for publication. But what we asked people if how they felt about words like invasive and sort of all that militaristic language that that surrounds invasion ecology and management, like eradicate and invasive, et cetera. And we're winning the battle. And the results were totally mixed. Some people thought this is really helpful and they felt very positively about it, and other people felt really negatively about it. And there were differences based on age, with younger people feeling that this militaristic metaphor surrounding invasions is really not productive and might steer them away from the field, whereas older respondents embraced it. And then there were some differences across ethnicity and race as well, with people who preferred not to identify having the most positive responses to it, and the most negative responses to the militaristic language, um, especially when combined with place-based common names like Japanese wireweed or Asian clam, etc., Chinese midden crab, those were most negatively felt by people who self-identified as Asian. So, I didn't ask specifically about Asian examples. That's just most of the examples out there. So, there apparently is a very strong reactions sometimes to these words, and with people responding that they felt threatened and excluded from the language and targeted by this language. So I'm trying. It's hard after decades of using invasive, et cetera, in European green crab, trying to shift away from that. Thank you for that question.
That's really interesting. We'll look forward to that publication for sure.
Thanks.
All right, I think we have run to our time, and I want to get folks safely out of this building that's half in the dark. So thank you very much for everybody online. Thanks for being here. For everyone in the room, again, thank you so much. We'll start our seminar series again up in the new year, and everybody have a good holiday. And Kat, thank you so much for being here. Much appreciated.
You're welcome. Sorry I couldn't be there in person, and everyone safe travels home.
Okay, thanks everybody. We'll see you in the new year. Bye now. All right, Kat, thank you so much.
Thank you. Bye-bye.